Action potential arrives at the synaptic knob, causing the calcium channels to open and calcium to flow in.
Increased calcium causes synaptic vesicles filled with acetylcholine to fuse with the membrane. Acetylcholine is released from the synaptic knob by exocytosis into the synaptic clip. Acetylcholine is picked up by receptors on the motor end plate of the skeletal muscle. Sodium channels open, allowing sodium ions to enter the muscle fibre.
Acetylcholinesterase in the synaptic cleft breaks down acetylcholine to end the signal.
The increase in sodium ions causes an action potential along the sarcolemma. Calcium is stored by calsequestrin in the terminal cisternae of the sarcoplasmic reticulum. Action potential travels down the sarcolemma into the T tubules, which stimulates the release of calcium from the terminal cisternae of the sarcoplasmic reticulum. Calcium then floods over the thin filaments and binds to troponin, pulling tropomyosin off the active site of the actin. High-energy myosin heads leap to bind on the exposed active sites on the thin filaments, forming a cross bridge. The myosin head pivots to the lower energy state, releasing ADP and moving the thin filaments over the thick filaments. This is known as the power stroke. The mitochondria, which stores ATP, lies in rows throughout the muscle fiber, strategically close to the muscle proteins that use ATP during contraction. ATP binds to the myosin head, causing the head to break free from the active site of the actin. The myosin head becomes energized as ATP is converted to ADP. Calcium is then pumped back into the sarcoplasmic reticulum and attached to the calsequestrin, allowing tropomyosin to once again cover the active site, resulting in relaxation of the muscle.
Myofibril creates the myofilaments, which is in the sarcomere. Z-discs separate one sarcomere from the next. The A-band is the darker middle part of the sarcomere where thick and thin filaments lie side by side. This is the zone of overlap. The I-band is a lighter area that contains only thin filaments and no thick filaments. The H-zone is the center of each A-band and contains thick but no thin filaments.
When the muscle contracts, the filaments slide over each other. Muscle relaxes, the filaments slide back towards their normal position.